Have you ever wondered what secrets lie hidden within the genes of an aneurysm and how understanding them could transform the way we treat these life altering conditions? Exploring the transcriptome of intracranial aneurysms is like unlocking a molecular blueprint, holding the promise to revolutionize treatments and bring precision medicine to the forefront of neurosurgery. I'm Sneha Sai Mannam, third year medical student at the University of Pennsylvania, and I will be discussing transcriptomics of intracranial aneurysms. I'd like to acknowledge that I used AI-generated images to add vibrancy and a modern touch to these slides. And to help distinguish these, each slide featuring AI-generated content will include the citation image generated by ChatGPT. This reflects the growing role of AI in helping us communicate ideas visually, and while we celebrate these advances, it's important to engage with them thoughtfully and responsibly. We're in an incredible era where technology and human creativity intersect, and it's a privilege to explore what AI has to offer. Transcriptomics involves studying the entire transcriptome, the full set of RNA molecules expressed by a cell, and to understand active gene expression. Over the past 2 decades, transcriptomics has rapidly progressed, establishing itself as essential to biological research and clinical applications. Now regularly applied to complex disease research, transcriptomics enables scientists to map gene interactions and identify biomarkers critical for diagnosing, treating, and understanding various conditions, including intracranial aneurysms. By examining gene expression changes, transcriptomics provides insights into intracranial aneurysm development, growth, rupture, and treatment response beyond what is visible in imaging or pathology. Transcriptomics allows for the identification of specific genes involved in aneurysm formation and progression, uncovering molecular processes previously inaccessible. This deeper understanding of intracranial aneurysms' molecular behavior holds the promise for targeted therapies, which could enable clinicians to predict intracranial aneurysm progression and customize treatment for individual patients. Flow diversion represents a significant advancement in intracranial aneurysm treatment, redirecting blood flow to promote vessel healing and aneurysm stability. Flow diversion has gained popularity due to its efficacy and safety, but our understanding of the underlying biological responses has not kept up with its clinical adoption. Despite extensive clinical use, relatively few studies have explored the molecular changes that contribute to flow diversion's effectiveness or potential complications. Transcriptomics offers a way to uncover the molecular changes that occur in response to flow diversion, revealing why some aneurysms respond positively while others may not. By analyzing gene expression changes in response to flow diversion, transcriptomics may help identify specific genes that indicate a successful healing process or predict potential treatment failures. Transcriptomic insights could one day enable personalized predictions of flow diversion outcomes, helping clinicians tailor treatments to optimize safety and effectiveness for each patient. Transcriptomics allows us to identify genetic expressions that are indicative of successful healing, potentially spotlighting key genes that promote or inhibit healing within the aneurysm. By identifying biomarkers in blood serum, transcriptomics could allow for non-invasive monitoring of flow diversion success and real-time tracking of treatment response. Combining transcriptomic data with proteomic and metabolomic information provides a fuller understanding of flow diversion's biological impact, facilitating more holistic research and advanced therapeutic insights. Transcriptomics explores the full transcriptome, or the set of all RNA molecules expressed within a cell, tissue, or organism, providing a real-time look into gene activity. The term transcriptome was first coined by Charles Offray, highlighting a new era in understanding cellular function at the RNA level. This field covers the full range of RNA biology, from transcription and expression levels to RNA locations, trafficking, and even modifications that impact gene function. Transcriptomics studies in individual RNA types, including mRNAs, microRNAs, and long non-coding RNAs, each playing distinct roles in cellular function. By analyzing transcripts, start sites, splicing patterns, and post-transcriptional modifications, transcriptomics reveals how genes are expressed and regulated. Transcriptomics also examines the locations, trafficking, and degradation of RNA within cells, offering a nuanced understanding of cellular regulation. The development of high-throughput sequencing technologies enabled large-scale RNA analysis, transforming transcriptomics into a precise and widely accessible field. RNA-seq provides a high-resolution look at the entire transcriptome within a cell, offering insights into gene expression levels and interactions. RNA-seq studies have deepened our understanding of intracranial aneurysm pathogenesis by identifying key genes and pathways involved in intracranial aneurysm development and rupture risk. RNA-seq has helped identify pathways in intracranial aneurysms related to stress response, metabolism, immune activation, and the extracellular matrix, all contributing to aneurysm formation. RNA-seq analysis reveals that processes like endoplasmic reticulum stress, autophagy, and leukocyte migration play roles in intracranial aneurysm pathology. Studies show distinct gene expression patterns that contribute to intracranial aneurysm vulnerability, rupture risk, and treatment responses. While RNA-seq provides a comprehensive transcriptome profile, it captures an averaged view across mixed cell populations, masking individual cell behaviors. RNA-seq doesn't provide details at the single-cell level, limiting the understanding of specific cellular responses within aneurysm microenvironments. To overcome these limitations, researchers seek more precise tools to capture the unique gene expressions of individual cells within intracranial aneurysms. Single-cell RNA-seq offers single-cell resolution, enabling researchers to profile the RNA expressions of individual cells and understand unique cellular behaviors. By examining gene expression at the single-cell level, single-cell RNA-seq allows for detailed profiling of specific cell subtypes such as macrophages, smooth muscle cells, and endothelial cells within aneurysms. Advanced clustering algorithms applied in single-cell RNA-seq help categorize cell types by their gene expression, offering a precise view of cellular roles in intracranial aneurysm pathogenesis and response to treatments. Single-cell RNA-seq has identified unique immune and vascular cell subtypes in aneurysm walls, showing how each contribute to disease progression and stability. Studies using single-cell RNA-seq reveal differential gene expression patterns across individual cells, which can help predict aneurysm behavior and guide targeted therapies. Single-cell RNA-seq's application in intracranial aneurysm research holds promise for developing precise treatments by targeting specific cell subtypes that play critical roles in intracranial aneurysm stability and rupture risk. Transcriptomic analysis of aneurysms uses 1 neurosurgeon-accessed clinical specimens, 2 bioinformatics analyses of existing genomic data, and 3 analysis of readily available clinical tissue samples such as blood. Single centers or consortiums can analyze transcriptomes from microsurgically obtained aneurysm domes, comparing these with control arteries like the superficial temporal artery. By using advanced bioinformatics, researchers can reanalyze existing data in novel ways, which adds depth to our understanding without additional invasive procedures with emerging dry lab analyses. The Gene Expression Omnibus, or GEO database, provides access to high-throughput gene expression data, including microarray and RNA-seq experiments, and allows exploration of genetic changes linked to aneurysm growth and rupture. Studies using GEO data mimic meta-analyses by combining small sample sets from various sources, unveiling new findings that would be missed in single studies. Liu et al. Used the GEO database to show concurrent hyperinflammation and immune suppression in aneurysm walls, which revealed imbalanced immune responses that contribute to intracranial aneurysm pathogenesis. This was then applied to a hybrid machine learning model using 4 GEO datasets to identify a 10-gene signature highly predictive of intracranial aneurysm rupture. The machine learning approach achieved an accuracy of 91.9% with an area under the curve of 0.94, showing strong predictive value. Although only one gene, GZMK, had previously been associated with intracranial aneurysms, this approach suggests machine learning's potential to uncover novel biomarkers. Beyond mRNA, transcriptomic studies have extended to serum microRNAs and circular non-coding RNAs, which can serve as valuable biomarkers for intracranial aneurysm development and treatment response. By analyzing microRNA and circular RNA expressions, researchers can identify unique expression patterns linked to intracranial aneurysm formation, rupture risk, and aneurysm inflammation. These RNAs are more accessible and stable in blood, making them promising candidates for non-invasive biomarkers through liquid biopsy approaches. Liquid biopsy involves studying altered gene expressions in circulating blood cells affected by intracranial aneurysm tissue, providing a non-invasive approach to transcriptomic analysis. Poppenberg et al. Found that a set of 18 altered genes in blood cells could diagnose intracranial aneurysms with 85% accuracy, highlighting key processes like leukocyte activation, macrophage recruitment, and inflammation. Ongoing research aims to apply liquid biopsy to predict aneurysm growth and rupture risk, integrating gene expression data with clinical scoring systems. Endovascular biopsy collects cells from aneurysm walls using coils, which offers direct access to aneurysm tissue for transcriptomic analysis. Cook et al. Used single-cell RNA-seq on endovascular biopsy samples to identify gene expression changes linked to aneurysm progression. Although this study demonstrated feasibility, it fell short of defining clear gene signatures for aneurysm rupture, which underlines the value of contributing this data to larger databases. RNA-seq data requires advanced computational techniques for analysis, which often blends scientific rigor with artistic interpretation due to the data's complexity. Unlike other genomics fields, RNA-seq still lacks standard practices for reproducibility, with issues like accurate clustering and significance adjustments needing refinement. Effective transcriptomic studies benefit from team-based approaches that include computational specialists to enhance accuracy and interpretability of findings. Unlike other aneurysm treatments, flow diversion involves multiple vascular regions, including the aneurysm dome, the parent artery, branch vessels, and the aneurysm neck, which forms a unique flow diversion microenvironment. For flow diversion to be effective, each region plays a critical role in device apposition and healing, with successful bridging of the device across healthy endothelium. By studying the transcriptome of each zone, researchers aim to enhance device compatibility while reducing thrombosis risk and promoting healing across a wide range of aneurysms. Modern next-generation sequencing technology allows for faster, more accurate, and cost-effective gene sequencing, which was previously limited to single aneurysm regions like the dome. Next-generation sequencing enables researchers to study gene expression across the entire flow diversion microenvironment, including the aneurysm dome, neck, and surrounding vessels. The accessibility of next-generation sequencing allows for comprehensive transcriptomic studies that were previously too costly or complex, offering a holistic view of flow diversion's biological effects. The aneurysm dome is the traditional focus for flow diversion research, where thrombus formation begins, but it isn't the only relevant area for flow diversion success. The proximal and distal ends of the flow diverting device must bridge healthy endothelium to ensure stable healing and minimize complications. Parent artery and branch vessels impact flow diversion efficacy as well, especially in complex anatomies, which emphasizes the importance of a device that integrates well with all microenvironment zones. Various surface modifications such as phosphocholine, heparin fibrin nanocoating, and glycan-based hydrophilic polymers are being tested to reduce thrombogenicity and improve healing. Technologies like CD31 mimetic coatings and bioresorbable devices show promise for facilitating endothelialization and reducing complications. By understanding gene expression across the microenvironment, these designs can be fine-tuned to create flow diversion devices that are compatible with complex aneurysm geometries and branch vessels. Adjunctive therapies such as stem cells or pharmacologic agents may enhance flow diversion treatment by promoting healing within the flow diversion microenvironment. Transcriptomics can reveal how these adjuncts interact with flow diverting devices at a molecular level, highlighting specific genes or pathways they influence. Studying adjunctive therapies within flow diversion microenvironments opens possibilities for multimodal treatment strategies, especially for challenging aneurysms. Studying how aneurysms and flow diverting devices interact in each microenvironment zone may address knowledge gaps regarding flow diversion treatment effectiveness. Multi-zone transcriptomic analysis can offer insights into flow diversion's limitations and opportunities for device optimization, especially in distal tissue perfusion and branch vessel effects. This approach could drive innovations that make flow diversion safer and more effective for a broader patient population, addressing unmet needs in intracranial aneurysm treatment. In conclusion, transcriptomics is a rapidly evolving tool with transformative potential in understanding and improving flow diversion and intracranial aneurysm treatments. Combining findings across clinical studies, preclinical models, and flow diversion microenvironment analysis will deepen our understanding of effective aneurysm management. Continued advances in transcriptomics could lead to safer, more personalized, and effective therapies, representing a new era in the management of intracranial aneurysms. I'd also like to acknowledge my PI, Dr. Srinivasan, who is also the lead author of this study. He allowed me to work on this project as a part of the Neurovert Clinical Research Laboratory at the University of Pennsylvania. And I also wanted to thank Dr. Stephen Brem. He's the director for the Neurosurgery Medical Student Education Program at the University of Pennsylvania, and was great in providing me with a great immersive experience in neurosurgery during my 2-week rotation as a part of my surgery block. And I included the collaborators for our study as well. And this is my contact if anyone is interested in discussing this further or for collaboration opportunities, and my email is right there. I also have a podcast titled Strive, Exploring Mind, Medicine, and Motivation if you want to check it out. And these are my references. Thank you.